Hi guys, so today our topic is about kinetics of unwinding of the double helix, and this will be by the thermal impact method. Okay, so the solution of DNA was, if we take a solution of DNA and it was heated from 5 to 20 degrees Celsius for 0.5 seconds, the kinetic curves indicate the presence of the last two steps and the structure transition. So in the first step, rapid step, almost all the hypochromic effects will disappear, and presumably the second stage will be the formed fixed loops, which will fully unwound at the second stage, which I was talking about. Sorry. And the rate of unwinding obeys the Arrhenius equation, and the energy of activation strongly depends on the pH. Okay, and the pH attaining a maximum value of the intermediate values of it. And an experimental investigation of the kinetics unwinding enables one to determine effects in the second structure of DNA. For this purpose, Lazarkin and his co-workers have worked out the kinetic formaldehyde method, which we call as KFM. Okay, so if the solution contains a substance which reacts with partially deparalyzed nucleotides, which thereby hinders the formation of Watson Crick pairs by these nucleotides, the reaction will then proceed into complete disparallelization, and it is attained at the time of binding of such reagent exceeds the unwinding time. And in this reaction, it is exactly one that limits the rate of unwinding the rate of constant DNA and reagent K, which will be K-W. So where W will be the probability of separation of any pair of nucleotides in the molecule, and K dash will be the constant, the rate constant of the reaction of separation of separated nucleotides with the reagent. And if some of the bases of native DNA have reached the formed a locally denatured portion, the probability will increase. The probability of thermal denaturation of the adjacent pairs will increase by W dash very greater than W. So the rate constant K dash is accordingly much greater than K. The formation of a locally denatured segment requires the free energy G, and therefore K by K dash will be equal to W by W dash, which is approximately equal to exp minus GS by RT, and which is equal to sigma, and which is approximately 10 raised to power minus 4 to 10 raised to power minus 5. Since K is less than K dash, so the unwinding process, which proceeds slowly, is gradually accelerated, and the curve showed the dependence of 1 by theta on time of the sigmoidal 1. And if the double helix has impact regions or originally disparallelized sections, the initial slope of curve will be increased. The curve, I'll show the curve. It will be like this. Okay, so here will be like 1 by theta, and 1.0 will write here, and here it will be 0, and here it will be 100, and here it will be 200, which is the temperature. Okay, so the curve will be like this till 1. It will be like this, and then it will remain constant. So this curve with the experiment of mononucleotides, it follows that the rate constant K dash of the reaction between the base and the formaldehyde is of the order of 1 per minute minus raised power minus 1, and hence K will be equal to WK dash, which is approximately lambda A K dash, and which will be 10 raised power minus 4 to 10 raised power minus 5. Okay, so the value of y is unknown, but it is reasonable to put it equal to a few units, and ky will be approximately 10 raised power minus 4 minute minus 1. Okay, so this value lies between the sensitivity limit of the kinetic experiments and may be neglected as compared with the 2kc. And since k, which we already saw that W dash is greater than W, so K will be also like this. So when we saw K is much lesser than K, and C of the units is the order of the units. So we'll observe that Z is equal to 2K dash by C, and the constant K dash is found from experiments on fragmented DNA from the formula, and in such cases C is unknown, and the order of magnitude of K will be 2.5 minute minus 1, and the lowest of the Z that can be measured will be 3 into 10 raised power minus 4 minus 1. So in other words, the kinetic formaldehyde method allows us to determine the minimum concentration of the defects order of one of the defect per 10 raised power 4 nucleotide pairs. So this method is very sensitive, and it is used with success for the studying of DNA with the breaks caused by the ultraviolet radiation or enzymatic hydrolysis. So native DNA is found to be practically free from defects which disapprove the zigzag model with sharp breaks. KFM is applicable to the study of complexes of DNA with RNA polymerase and of the fine structure of DNA. So the denatured coil-like single-stranded chains of DNA are capable of the renaturation, and they can again form a double helix. The renaturation of DNA was discovered and studied in it was studied in 1961 by Marmer and Doughty. Okay, so slow cooling, that is annealing of denatured DNA, results in a partial recovery of the biological activity and hypochromic effect. So renaturation also occurs after rapid cooling, quenching of the denaturated DNA of the simple ages, and if it is again, it's subjected to heating up a temperature somewhat lower than Tm, and renaturation is easily followed from a change in density with the aid of sedimentation in the CSCl density gradient. And for instance, the density of native DNA, DNA will be 1.704 gram per centimeter cube, whereas the denatured and quenched in native DNA has density of 1.716 gram per centimeter cube, and the density of the denatured DNA is 1.700 gram per centimeter cube. So renaturation is generally dependent on temperature, and the optimum temperature for renaturation is high enough for unwinding the coils and is at the same time low enough for the double helix to be stabilized. And DNA from Diplococcus pneumoniae undergoes renaturation at 67 degrees Celsius and at the rate of 2.5 times faster at 80 and 50 degrees Celsius. So renaturation process is readily in case of phase DNA with some difficulty of bacterial DNA, and is practically infeasible for the samples produced from calf thymus. Cells of E. coli and phage T2 had a molecular weight of 10 raised power 7, and the number of various DNA molecules per cell or phage particle would be 10 raised power 6 or 10 raised power 2 and 10 respectively. With the same weight concentration with all the samples would have quite different concentrations of complementary chains, and the probability of encounter of two such chains formed on denaturation of DNA in the sample produced from the cells or higher organism will be very low, and renaturation can lead to the formation of hybrid duplexes from DNA's virus origins, and they have been obtained hybrids of DNA isolated from various strains of E. coli, and also interspecies bacterial hybrids, and hybridization of DNA disclosed the evolution genetic relations between bacteria. That's all. Thank you.